Welcome to the Sans Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Before we dive in, please take a moment to like this video and subscribe to our channel. It really helps support the Sans Science Experience. Okay, so today we're cutting through the noise on a truly, well, I think it's a groundbreaking idea, cellular aging. You know how different parts of us seem to age differently, like hair, heart, even how we think. They don't all follow the same clock.
Exactly. It's something we observe, but the why at the cellular level is fascinating.
Right. And our sources, especially this paper by Ming Yang and colleagues, they really dig into that why.
Yeah.
So the mission for this deep dive is to figure out how the lifespan of individual cells explains this asynchronous aging we see in tissues and organs.
And there's some pretty surprising insights, suggesting our body isn't just one aging entity, but more like a mosaic of different cellular timelines. It challenges that idea of a single biological clock.
Okay, let's unpack this core concept then. Cellular age. The research suggests, basically, that how long a cell has actually been around, it dictates how much its gene expression gets, well, noisy.
Yeah, noisy is a good way to put it. Less precise, more variation in gene activity. Think of a clear signal becoming fuzzy over time.
And the link they found is pretty direct, isn't it?
Astonishingly direct. They looked at cells from tissues that don't renew very quickly, like neurons or muscle cells. These can be almost as old as the organism itself. And using mouse single-cell data, they found this really striking positive link. The older the cell type, the more variation, the more noise in its gene expression.
Wow.
Yeah, get this. In 24-month-old mice, which is quite old for a mouse, the cellular age alone explained fully half of the variation they saw in gene expression patterns.
Half, just from the cell's age.
Half. It's huge. It suggests the cell's, birth date is incredibly critical to how it functions later on, maybe as much as its immediate condition.
That definitely challenges how we usually think about it. So older cells get noisier, but what about cells that don't stick around, like blood cells or gut lining cells? They turn over constantly.
Right, great question. Do they just get replaced before the noise kicks in, or is something else going on? Well, the research uncovered this really interesting contrast in strategies. The long-lived cells, like in the heart or brain, they seem to invest heavily in what's called proteostasis.
Proteostasis?
Yeah.
Like protein stability.
Exactly. Think of it as advanced internal housekeeping, making sure proteins are folded right, clearing out damaged bits, even keeping the mitochondria, the powerhouses, tidy. They are essentially built to last, investing in self-maintenance.
Okay, so they focus on preserving themselves.
Right. But then you look at the short-lived cells, the ones turning over every few days, and counterintuitively, they actually express more genes involved in DNA repair.
More DNA repair. Why, if they're just going to be replaced anyway?
Well, it points to a different strategy altogether. They might inherit some wear and tear, some damage from the stem cells they come from. But because they turn over so fast, any cell that accumulates too much damage just gets weeded out and replaced by a fresh young one very quickly.
Ah, I see. So it's less about fixing individual cells long-term and more about constantly refreshing the pool.
Precisely. It's like a systemic renewal mechanism. The organism benefits because damaged units are efficiently removed rather than trying to patch every single one up indefinitely.
So it's really not a one-size-fits-all approach to aging in the body. It's more like this dynamic balance. Some cells focus on self-preservation, others rely on rapid turnover.
Exactly. A balance between intrinsic maintenance and systemic renewal.
That definitely flips the script on the idea of a single aging process. What I find kind of mind-blowing here is this idea of cellular demography, our body as a mosaic of different aging strategies all happening at once.
Yes. And if you take that bigger picture view, understanding these diverse strategies could really shift how we think about, say, extending human health span.
How so?
Well, instead of searching for that one magic bullet, that universal anti-aging fix, we might need to think about cell type specific approaches. Interventions that actually boost the natural aging strategy of particular cells, supporting proteostasis in neurons maybe, or ensuring efficient turnover in the gut.
Tailored interventions based on how different cells are already designed to age. That makes a lot of sense. So for you listening, consider this. If our cells really are designed to age in these different ways, what kind of revolutionary insights could we unlock if we start embracing that diversity? How could that change our pursuit of longer, healthier lives? That's it for this deep dive. Don't forget to like this video and subscribe to the Sans Science Experience for more deep dives into the latest science.